We're going to discuss Taenia saginata and Taenia solium egg. It is important for you to take note that the egg of Taenia saginata and Taenia solium is indistinguishable to one another. If you still remember our discussion in hookworm, no? The hookworm egg is also indistinguishable to each other. That's why if you see the presence of the 8 embryonic cleavage, no, with a thin outer cell wall, that is your hookworm. No? And if you report the presence of hookworm egg in the stool, means it can be Ancylostoma duodenale or Necator americanus. The same thing goes with Taenia saginata and Taenia solium. Okay? The same thing goes with Taenia saginata and Taenia solium, wherein you cannot identify specifically if that is saginata or that is a solium, unless otherwise you are going to measure the egg. If you're going to take a look at a glance, you might say that the egg of Taenia saginata is almost similar to the structure of the egg of Ascaris lumbricoides, most especially if the egg of Ascaris lumbricoides is decorticated. However, don't be confused and don't be easily deceived. Why? Because there is a distinct characteristic of the egg of Taenia saginata that can only be seen predominantly to Taenia saginata and Taenia solium, and that is the presence of the radial striations. There's also 3 pairs of hooklets, and of course the hexagant embryo. That is unique to Taenia saginata. Meron siyang radial striation, Taenia solium and Taenia saginata, or Taenia egg, Taenia species. That's why if you will be able to see the presence of this egg in a sample, you cannot say that it's saginata. You cannot say that it's solium, because you have to. Thus, therefore, if you see the presence of this egg, you're going to report it as the presence of Taenia species. Unless otherwise, you are going to see the presence of the Scolex. Again, the Scolex of Taenia saginata and Taenia solium will help us to differentiate between the two tapeworm. One is the presence and the absence of hooklets, well, of course, the presence and the absence of the rostulum. If you don't have the rostulum, then you are saginata. If you have the rostulum, then you are solium. Rostulum, solium. Sounds like, di ba? If you don't have the rostulum, you are saginata. But if you have the rostulum, you are solium. And take note, dagdagan na rin din natin, if you have the presence of rostulum, since that you are a Taenia, kapag mayroong rostulum, meron siyang hooklets, that is your Taenia solium. Next, in terms of similarities, they have the same number of the suckers or the ventral suckers. Okay, quadrated. Okay, 4 ventral suckers for both saginata and solium. All right. Another thing that will help us to differentiate solium from saginata is their proglottids or the body. No? For Taenia saginata, we have what we call the uterine branches. And these uterine branches are sometimes referred as the lateral branches on each side of the proglottids will help us to differentiate whether it is a saginata or solium. Number 1, in terms of the number. Okay. In terms of the number of the uterine branches or the lateral branches, saginata, okay, is 15 to 30, while solium is 7 to 5. Thus, therefore, mas marami pong uterine branches ang ating saginata as compared to our solium, wherein there are only 7 to 5. In terms of shape, the proglottids of our Taenia saginata is somehow longer but narrower than the proglottids of Taenia solium. Si Taenia solium somewhat square. Okay, maikli pero medyo may katabaan or medyo may malaparan. Another thing that will help us to differentiate is the number of proglottids that can be seen in an adult worm. However, we cannot count naman yung proglottids. Ang hirap to, no? Ang hirap i-count ng proglottids nila. Okay? It can be useful to differentiate, no? Taenia saginata, but it is inappropriate to do that in the laboratory. Eh, paano kung naputol lang yun? Diba? And since this is segment, hindi mo magagawa yun. But in terms of the number of proglottids, take note of this, guys. Taenia saginata can have up to 2,000 proglottids. Could you imagine? A single adult Taenia saginata could have 1,000 to 2,000 proglottids. While for Taenia solium, we have less than 1,000. Okay, sabi sa book, average of 848 at ito daw ay 1,048. The laboratory diagnosis for this one is the findings of the egg and the gravid proglottids in the stool. However, no, Scolex may also be seen, but in cases that the parasite is dead. And we said that a successful treatment, a successful medication, a successful management against Cestodes will demonstrate the presence of the Scolex. Once the Scolex is still attached in the mucosa, it can still cause infection. Perineal opening using cellophane tape swab or tape rep procedure has resulted in the recovery rate of the parasite. So recovery of the egg or recovery of the proglottids. In terms of the life cycle, okay, the life cycle of Taenia solium and Taenia saginata are almost the same. They just differ in terms of its intermediate host. The intermediate host of Taenia saginata is the cattle, the, the cow, no? And the intermediate host of the solium is the pig or the swine. The infection initiates, or the initiation of the infection occurs when a person ingests a contaminated pork or contaminated beef with the Cysticercosis larvae. Okay? Ingestion of contaminated pork or contaminated beef. I want to make it clear. Sometimes students got confused every time that I ask, what is the infective stage or what is the mode of transmission of Taenia saginata and Taenia solium? They are always keep on telling and keep on saying that the the mode of transmission of Taenia saginata is the ingestion of cow. Who could you? O, natawa ka dyan, di ba? Nakakatawa. Ingestion of a cow. Eh, mas malaki pa yung baka sa'yo, kakainin mo yun ng buo. Di ba? It is right and appropriate to say ingestion of contaminated beef, which is the meat of the cow, instead of saying ingestion of cow. Cow or cattle is the intermediate host, but it doesn't necessarily mean that you have to ingest the cow. The same thing goes with pig. The same thing goes with solium. You cannot say that the mode of transmission of Taenia solium is the ingestion of pig. Mantokwang pating tayo dyan. Okay? Let's correct the ingestion of contaminated or raw. Ingestion of contaminated, raw, or undercooked meat or beef or pork, no? Pork or beef. Kaya nga pork tapeworm and beef tapeworm. Ang sarap magsangyup. Kaso pandemic. Ibig sabihin, medyo safe ka. Is it possible? Yes, it could be. Sure, lalo na kung hindi naman naluto ng husay. Di ba? Konting usok-usok lang, gutom ka na, di kain ka na agad. Something like that, okay? So ingestion of contaminated raw or undercooked pork or undercooked beef. Kaya mga nagalabat, luto na luto. Okay? Yung baboy at ang baka. Now, how about the infective stage? This contaminated pork, this contaminated beef contains the Cysticercosis larvae. Okay? Ano ba yan? Mali spelling natin. Baka sabihin kaya mali yung sulat ko. I've been using this pen for almost a year, but I still don't know how to write good. Cysticercosis. Yan. Ay, wala akong S. Cysticercus. Cysticercus. Okay? Cysticercus larva is the infective stage that is seen in the meat, no, of either pig or cow. For saginata, specifically, saginata. For T. saginata, we have Cysticercosis bovis. Bovine ata ang sabi ko kanina. Bovis po yan. No? Cysticercosis bovis. And for Taenia solium, you have your Cysticercosis cellucei. Yan. No? These are the infective stage. So pag tinanong kayo, what is the infective stage of Taenia saginata? Okay? Cysticercosis bovis. And you can get that type of infection if you accidentally ingest a contaminated beef. Okay? Not a contaminated cow. Not a contaminated pig. Now, after the ingestion of a meat that is contaminated with bovis and cellucei, bovis or bovine, it will mature, no, into the humans. Then the Scolex will attach to the intestine so that it will be able to produce segments, proglottids, to have its robilla. Remember, they are hermaphrodite. Even without the presence of their partner, they can reproduce. Okay? So there will be the production of egg that can be passed through feces, no? Na pwedeng nandoon sa, most especially in rural areas or in proper or inappropriate disposal of stool, if predominant, pwedeng makain ng baboy or pwedeng makain ng yung stool ng patient with the egg of the parasite. Now, I want you to take note that Taenia solium can cause what we call cysticercosis that is possibly seen in the brain or in the muscle. It is not confined or it is not limited only in the intestine, but also capable of invading the brain, cerebral cysticercosis, okay, which is considered as one of a serious complications of Taenia solium. Okay, so when we said Taeniasis, that is the infection brought by Taenia saginata or Taenia solium. And there is a non-descriptive symptoms like diarrhea, abdominal pain, weight loss, slight weight loss, or change in appetite, dizziness, vomiting, nausea, and of course, moderate eosinophilia. Prognosis is usually good. Okay naman, nagagamot. How about cysticercosis? Cysticercosis is the involvement of the muscle, brain, heart, lung, and peritoneum, which is capable of producing inflammation. Thus, therefore, symptomatology is dependent on the number of the parasite and, of course, the location of the parasite. If there is involvement of the brain, then symptoms may include uh, loss of confusion or loss of uh, loss of dito? Yeah, I can do it. I can do it. Loss of loss of yeah, loss of mind. Ang nangyari kay sir? Unconsciousness, okay? Could be or mental convulsions, something like that. For striated muscles or pain in the area where the parasite is present. Now, if in case that there is an involvement of the brain, it is what we call the cerebral cysticercosis, or usually or commonly referred as the neurocysticercosis. Neurocysticercosis is a serious infection. Okay, this is a serious infection that is usually seen worldwide. Actually, this is a zoonotic disease. Kasi parasite of pig naman to eh. Neurocysticercosis is uh, what's it called this? Neurocysticercosis is common to a parasite na Taenia. Okay? Eh yung parasite na yun, ay parasite ng mga baboy, pero kinain mo yung contaminated pork, nakameron ka ng infection. Okay? The death of the larvae leads into the inflammation of the affected region. This is an example. I don't know if you love to see this picture, but I myself will close my eyes because I don't want to see this picture. Bahala na lang kayo if you want, or you just want to cover. If you see the picture, or if you are looking at the picture, as you can see, that is a biopsy of a brain or cross section of brain. Again, uh, and the holes that you can see is the site where the parasite invades. Looking at this CT scan, I'm having goosebumps. Looking at this CT scan, can I skip? Yeah. Looking at sorry, ako na lang pala. Looking at this CT scan, as you can see, there are holes, no? This represents the site of invasion of the parasite. And this sample or this area is useful for biopsy procedures. CSF can also be used as a sample of choice for the detection of cysticercosis, or particularly the neurocysticercosis. Okay, and once that you have a lumbar puncture between the fourth or the fifth lumbar vertebrae, merong mataas na pressure, elevated ang protein, mababa ang glucose, and increased ang mononuclear cells. Tapusin na natin, parang awa na lang. Okay, that is neurocysticercosis caused by Taenia solium. The drug of choice is no other than Praziquantel. This is common to all cestode and rheumatoid infection. Praziquantel is the drug of choice. Alternative treatments such as paranamycin and quinacrine hydrochloride can also be used. Leading to the third parasite, the Taenia asiatica, or sometimes referred as the third Taenia species, usually referred as the Taiwan Taenia, because it was first described or discovered in Taiwan. Thus, it is referred to be the Taiwan Taenia, and also called the Taenia saginata because of its characteristics that is almost similar to Taenia saginata. Okay? It can also be seen in countries such as Korea, China, Philippines, Thailand, Malaysia, and Indonesia. Morphologically similar to Taenia saginata, but the Scolex is almost similar to Taenia solium. Thus, if that is similar to Taenia solium, therefore, there is also the presence of perostulum. Now, in terms of intermediate host, it includes cattle, goats, monkeys, swine, and wild boars. I will always remember and will never forget this question in our board exam. The question goes like this. Which of the following is considered as an intermediate host of Taenia asiatica? Okay, again, the question goes like this. Which of the following is considered as the intermediate host of Taenia asiatica? Okay. Me, who knows that cattle, cows, monkeys, swine, and wild boars are considered as the infective stage. Hindi po ako natakot sagutin ang tanong. But what makes me feel nervous is the choices. Why? The choices are A. Cattle, goat, monkey. B. Swine, monkey, wild boars. C. Wild boars, goat, monkey. E. Monkey, swine, goat. Ano? Ang sasagot mo? And my students are always asking me, ano po ang sinagot mo, sir? Okay? Ang sinagot ko ay yung monkey, swine, and wild boars. Tama po ba kayo? Hindi ko rin alam. But one thing I know, pumasa ako ng board exam. Okay? Whatever man tanongin yun, hindi ko alam kung ano ang sagot. But knowing that all of those are considered as intermediate host. So if you will ask, bakit yun ang sinagot ko? Bakit monkey, swine, and wild boars? Bakit hindi yung goat? Bakit hindi yung cattle? Bakit yung monkey, swine, and wild boars nga? Number one of my reason is that monkey and wild boars. That is the most common intermediate host of Taenia asiatica, monkeys and wild boars. But I still don't know if I got the correct answer on that particular question. But to rationalize my answer is that because of monkey and the wild boars. Yung mga baboy ramo. Okay? The cysticercosis of this parasite is usually seen in the liver. Moving forward to our fourth parasite, which is Hymenolepis diminuta. Okay, again, ito naman yung tanong sa amin, o board exam, sa Hymenolepis diminuta. The question goes like this. How are you going to describe the egg of Hymenolepis diminuta? Hindi mo pwedeng sabihin doon na circular lang siya with the presence of the polar thickenings and embryophore. Okay. You have to describe the egg of Taenia diminuta. Hymenolepis diminuta. Inantok na. Okay, commercial lang yun. Hymenolepis diminuta. Ano yan? Take note. This is how are you going to describe the egg of Hymenolepis diminuta. 55 to 85 micrometers in size, with the presence of 3 hooklets and hexicant embryo. There is a distinct polar thickening. Agad. There is a distinct polar thickenings. Okay? The distinct polar thickenings, colorless embryophore, and no polar filaments. Why I am asking you to remember this? Because the egg of the diminuta is almost similar to the egg of Nena. Oh, the egg of the diminuta is almost similar to the egg of Nena. Later, we'll discover the difference between diminuta and Nena. But then again, remember, hexicant embryo, 3 pairs of hooklets, distinct polar thickenings, colorless embryo with no polar filaments. This is the head or the scolex of Taenia diminuta with a rostulum with no hooklets. The proglottids contains a sac-like uterus which is filled with eggs. The number of suckers is 4 since this is a cyclophilidian quadrated. A rostulum is present but no hooklets. Okay. The laboratory diagnosis for this one is of course the stool. Now for the life cycle, take note guys that typically this is a parasite of rats. Okay? Pero hindi si rats yung ating hindi si rats yung ating intermediate host. Rat is considered as the natural host of Hymenolepis diminuta, but hindi po si rat ang ating intermediate host. The intermediate host of diminuta is the fleece beetle. Okay. The grain beetles and the fleas are considered as the intermediate host of the parasite. Therefore, ingestion of fleas, ingestion of grain beetle is considered as the initiator of the infection. And the development no, of its adult form occurs into the man. Kaya nga definitive host tayo, accidental host, because we allow the development of the adult stage. Therefore, no ingestion of this fleas, ingestion of this grain beetle, which is considered as the intermediate host. Why? Because it allows no, the development of the cysticercoid larva of our diminuta. Therefore, ingestion of this no, will initiate the infection. So again, the scolex no, with one rostulum with 4 hooklets, and the body no, and the sac-like structure contains the egg of the parasite, can be seen in the small intestine of a person with this type of parasite. Therefore, the eggs will be passed again to the stool and to the environment. So ingested by arthropod, intermediate host, and then oncosphere hatch and penetrates the intestinal wall of the intermediate host or the arthropod intermediate host. Again, rat is considered as the the natural host, okay, of the parasite. Clinically asymptomatic, but in cases of symptomatic infection, the patient will experience diarrhea, nausea, abdominal pain, and anorexia. Again, take note, guys, that Hymenolepis diminuta is known to be a rat tick. Again, the drug of choice is so available as well. Next in line is your Hymenolepis vena, considered as the dwarf tapeworm. Typically, no, at a glance, the egg of Hymenolepis taenia is almost similar to the egg of diminuta. There is hexagon embryo, there is hooklets, tatlong hooklets din. No? Meron din siyang polar thickenings, at meron din siyang colorless embryophore. But what makes diminuta different from Taenia is the presence of 4 to 8 polar filaments. Take note, the presence of the 4 to 8 polar filaments in the egg of Taenia makes it different from diminuta. Then of course the size will do. But again, if your microscope is not capable of measuring the size, therefore we have to remember that if there is a polar filaments like this, okay, that is your Taenia. In terms of the head, almost similar. Yun nga lang, ano yung meron kay Nina na wala si diminuta. Si Nina, meron siyang hooks. Si diminuta, meron siyang rostrum with no hook. The proglottids of diminuta is almost similar to the proglottids of Nina. The size is twice as long, and there's again a sac-like structure filled with the eggs of Nina. Again, the laboratory diagnosis is the presence of the eggs in the stool. The life cycle, no, is almost similar to the life cycle of diminuta. However, it only differs in that Hymenolepis nana may or may not require insects as intermediate host. Thus, therefore, the infective stage of the parasite is ingestion of food contaminated with eggs of the parasite. In some cases, or in some life cycle, it may require or it may involve the presence of the host, and that is an insect host. Okay? Si Nena ay parasite of the rats ulit. At nakakain si tao, si human, ng droppings ng rat that contains the egg of the parasite. That initiates the infection. Okay? Uh, it could be from the water, it could be from the food, or it can be to the hands of the parasite, hands of the host ng tao. Okay, so these are the symptoms: abdominal pain, anorexia, dizziness, and headache. For the treatment, it is no other than the presence of or the use of Praziquantel as the drug of choice. Alternative drug in the USA, alternative drug, however, it is not available in the USA, is nicotinamide. Prevention and control involves proper hygiene and sanitation practices and avoidance of contact with infected rodents' feces.